We get started. So today's seminar speaker, why there are more people dialing in, as I can see, is Dr. Jill Koshiolfrom, who received her PhD in epidemiology from the University of North Carolina at Chapel Hill. And she actually has been at the NCI, if I understand it correctly, since 2005 and now serves as a senior investigator in the infections and immunoepidemiology branch of the Division of Cancer Epidemiology and Prevention. And it's not a surprise for us, but she's actually very interested in hepatobiliary cancers. So her main focus are biliary tract cancers and gallbladder cancer. And she's been studying different cohorts. One cohort from Chile, and then there's others in the REVEAL study that obviously all the hepatologists know that are dealing with hepatitis B and C. I believe she's also been involved in a longitudinal study in Chile on biliary cancer. And I got to know her because of her and my interest in gallbladder cancer, which is obviously a very, I would say, still understudied topic. So with this, I don't want to take any more of your time. And I would like to ask everybody to keep their camera off, as you already have. And then once we finish, we will have a discussion, and I suggest that you actually say the discussions in person rather than going through a chat menu.
Great. Thank you so much, Tim, for that great introduction, and also just to the liver cancer program in general for giving me this opportunity. I'm really excited to share some of the work that we've been doing to try to better understand the epidemiologic and molecular characteristics of biliary tract cancers. I'd like to start today by giving you some general background and then talking about the Biliary Tract Cancer Pooling Project, just to show you how we can use large-scale pooling efforts to prospectively evaluate biliary tract cancer etiology. Then I'll talk about a novel association that we observed between aflatoxin and gallbladder cancer, and then go into work that we've recently completed to better understand the molecular characteristics, which has both helped us better understand aflatoxin and given us new insights into gallbladder cancer. And finally, I'll wrap up describing the Chile Biliary Longitudinal study, which is where I really see the future in terms of trying to address some of the outstanding gaps that we just haven't been able to get at for gallbladder cancer etiology. So starting with the background, biliary tract cancers include intrahepatic bile duct cancer, gallbladder cancer, extrahepatic bile duct cancer, and ampulla of Vater cancer. And I study all of these cancers, but a lot of my work has been focused on gallbladder cancer. And that's in part because I think that gallbladder cancer is a really good model for trying to understand how inflammation contributes to the development of cancer. That's in part because gallstones are the single most important risk factor that we know about. And as you can see from this picture, they create a lot of damage and inflammation in the gallbladder. But other proposed risk factors also have an inflammatory component. For example, bacterial infections like Salmonella typhi, and diabetes, obesity, and metabolic syndrome. Another thing that's unusual with respect to gallbladder cancer is that it's one of the few cancers that is more common in women than in men. But as Tim mentioned, this is a very understudied cancer, and that's in part because it's rare in much of the world, and especially in industrialized countries. Now here you can see the overall age-standardized incidence rate for gallbladder cancer in men and women. And what you notice is that although gallbladder cancer incidence is low in much of the world, with rates about 2 per 100,000 or less, there are hot spots where the burden of this cancer is really pretty high. For example, southern central Chile is one of these areas with incidence rates of about 16 per 100,000 in women and 5 per 100,000 in men. Rates are more intermediate in a place like Shanghai, China, and I'll be sharing data from Shanghai just a little bit later on. But that's still quite a bit higher than what you see in a low-risk country like the US. Now, that said, even in the US, there are about 12,000 new gallbladder and extrahepatic bile duct cancer cases every year and 4,000 deaths. And there are notable disparities in the burden of this disease, with higher incidence rates among Amerindian groups and Hispanics, as well as other non-Hispanic white racial ethnic groups. And despite that, really very little work has been done on this area, which means that there's a lot that we can contribute. As I mentioned, gallbladder cancer incidence is higher in men than in women, which you can see from these incidence rate ratios. So here we're comparing the incidence rate in females to that in males. And as you see, that incidence rate is above 1, therefore you can see the rate is higher in females than in males. And what I think is particularly interesting here is that that disparity between females and males is even greater among younger age individuals who are age 18 to 44, as opposed to those who are age 45 and above. And we see that same disparity occurring when we compare non-Hispanic blacks to whites, when we compare Hispanics to whites, and when we compare other races to whites. In this table, the estimated annual percent change, or APC, shows you that the gallbladder cancer incidence rates are actually decreasing across all racial ethnic groups, except among non-Hispanic blacks, where the rates are actually increasing. So to look at that in a little more detail, here you can see in this panel the rates among whites in the US, and you see that among females we see this nice decline. In males, the rates were declining but have actually leveled off a little bit in recent years. And looking among non-Hispanic blacks, you can see among females, this orange line, that the rates are actually increasing. But the rates in males are a little bit more unstable, so it's harder to say what exactly is going on in that group. It's been known for a long time that Native American groups are disproportionately affected by gallbladder cancer in the US, as shown by these classic studies, including Native Americans from New Mexico and from Arizona, where you can see that Native American males and females have higher gallbladder cancer incidence rates than other racial ethnic groups. And that holds true today. Both in females and in males, the proportion of biliary tract cancers due to gallbladder cancer is larger among American Indians and Alaskan Natives than any other racial ethnic group. Interestingly, that incident rate ratio for Native Americans compared to non-Hispanic whites varies depending on where you look in the country. Here you can see that Alaskan Natives and Native Americans in the Southwest have the highest incidence rates compared to whites. And just to drill down on that a little bit farther, giving a state-level example from the Southwest, you can see from Ramagulapalli's group the gallbladder cancer incidence rates by county in New Mexico for whites, for Hispanics, and for Native Americans. And again, as you can see, the highest rates are among Native Americans. Amerindians have been shown to have a disproportionately high proportion of gallstones, as indicated in this figure by the orange squares and lines, and also a higher rate of gallbladder cancer indicated by the pink bars. So what do we know about gallstones? First, as I mentioned, gallstones are the single most important risk factor that we know of for gallbladder cancer. They lead to a great deal of inflammation or chronic cholecystitis in the gallbladder, and then together with additional molecular changes, that can lead to intestinal metaplasia, dysplasia, carcinoma in situ, and ultimately invasive cancer. So based on this model for gallstone-related carcinogenesis, there are really two important pieces to think about. First, what increases your risk of developing gallstones? And second, once you've developed gallstones, what increases your risk of developing gallbladder cancer? Now, the clinical mantra for the major factors associated with gallstones is female, fat, fertile, and 40. And these same factors are associated with increased risk of gallbladder cancer. So it's sort of hard to tease apart whether these factors increase the risk of gallbladder cancer only because they increase the risk of gallstones, or whether they contribute something to the development of cancer that goes above and beyond any effect that they have on gallstones. And that's a really difficult puzzle to tease apart because the vast majority of gallbladder cancer patients have gallstones, whereas only about 10 to 20% of the general population has gallstones. So a key question is what drives the progression to gallbladder cancer among people who already have developed gallstones? And that's something that I'll come back to when we get to Chile Bills at the end of the talk. First, I would like to talk about the Biliary Tract Cancer Pooling Project, which is an important resource that I built together with Pete Campbell from the American Cancer Society and Catherine McGlynn, who's also with me in DCEG. And this is something that we built together so that we could prospectively study biliary tract cancers. This effort involves 30 different prospective studies from all over the world with 3 million participants and over 5,000 biliary tract cancer cases. Now, this resource is particularly important because there are very few data existing in the literature on the association of even classic cancer risk factors like smoking and how smoking is related to the risk of cancer across the entire biliary tract. And what data do exist are typically conflicting. So BitCap gives us, for the first time, the potential to prospectively evaluate these kinds of associations at each anatomic site in the biliary tract, really improving our understanding of the etiology of biliary tract cancers. Now here you can see the distribution of cancers, and as expected, the majority of gallbladder cancers are female, whereas there tends to be a male predominance for the other types of biliary tract cancers. But do keep in mind that this distribution is influenced, of course, by the composition of the cohorts themselves, and there are some cohorts that are female only or male only. So don't take this as the truth for the distribution of all biliary tract cancers, but it does show you what we have in BitCap, and that generally we see some of the characteristics that we'd expect to see from biliary tract cancers. You can see that the median age of diagnosis is 72 to 73 years. Now pulling back a little bit to look at all participants in BitCap, you can see that the median age is 58 years, that 61% are female, and that 78% are white. And that's something I want you to keep in mind. We'll come back to that shortly. Now, to give you a sense of how we've been using data from BitCap to inform our understanding of the etiology of biliary tract cancers, I want to give you just 2 examples of the studies that we've conducted. First, smoking is a great example of a classic cancer risk factor that BitCap allows us to prospectively evaluate with risk of cancer across the entire biliary tract. There have been a few previous studies, but they've typically been cross-sectional or quite small, and so they've produced very inconsistent results with everything from positive to inverse associations. So to address this knowledge gap, my former post-bac fellow, Emma McGee, who's now finishing up her PhD at Harvard, she used data from 26 prospective studies in BitCap with nearly 4,000 incident biliary tract cancer cases and about 39 million person-years of follow-up. Here you can see that current smoking was associated with about a 2-fold increased risk of extrahepatic and ampulla-Vater cancer. Intrahepatic bile duct cancer was associated, but more modestly, whereas we didn't see a whole lot of evidence of an association with gallbladder. To us, it really looked like it wasn't associated. There weren't consistent trends with amount or duration of smoking. And that's interesting because shortly after we published our results, a meta-analysis was published that reported that current smoking was associated with gallbladder cancer with a pooled estimate of 1.33. Now, this estimate does include both case control and cohort studies, and case control studies are a great place to start, but there's always some concern that there could be bias due to things like recall or reporting bias. In other words, cases may be more likely to remember or report accurately their smoking behaviors than controls, and that potentially could falsely elevate the estimates. So focusing for a minute on the cohort studies, I do want to point out that 4 out of the 10 estimates presented here are actually not specific to gallbladder cancer, but are for gallbladder plus bile duct cancers. And that is a problem because, as you just saw, the association depends on where you are in the biliary tract. So if you combine things, the estimate that you see is going to be skewed towards whichever cancer site predominates in that study. Now, that said, I do think that it's worth noting that most of these cohorts are from Japan and from Korea, where the risk of gallbladder cancer is higher than in many Western countries. And this comes back to the point I made earlier. The majority of the studies in BitCap are from North America or from Western Europe. So we have largely a Caucasian population, and these are low-risk populations. In addition, pancreatobiliary maljunction is more common in Japan and Korea. So it's possible that the risk factors just plain differ between these populations, and the effect of smoking potentially could differ in the context of these different risk factors. To summarize where we're at with smoking, one thing I didn't mention earlier is that we saw the same trends across the biliary tract for amount and duration of smoking in BitCap that we did for current smoking. And that suggests that these associations really do reflect true differences across the biliary tract, not just chance findings. The recent meta-analysis underscores the need for more studies in non-European and North American populations so that we can really try to get at this question of whether or not there is heterogeneity in different geographical areas. And we do need to do more work to try to understand the biology that might be driving these associations that we're seeing. For my second example in BitCap, I want to present our work on reproductive factors, which is led by my postdoctoral fellow, Sarah Jackson. We're interested in reproductive factors because, as I mentioned, we see this really striking disparity in the sex ratio across biliary tract cancers, where gallbladder cancers are more common in women than men. When you look worldwide, that's at about a 2-to-1 ratio, whereas there tends to be a male predominance for the other types of biliary tract cancers. We think it makes sense that hormones could affect the gallbladder because the gallbladder does express both estrogen and progesterone receptors, and also high circulating levels of female sex hormones have been shown in clinical trials to lead to the development of gallstones, which, as I mentioned, is an important risk factor for gallbladder cancer. So we evaluated female reproductive factors as a proxy for sex hormones in BitCap. And in this panel on the left, you can see that increasing number of live births was associated with gallbladder cancer and with intrahepatic bile duct cancer, but not with extrahepatic bile duct cancer or ampulla of Vater cancer. And just to point out, these hazard ratios are per live birth. So they may look small, but when you add up live births, they actually become quite large. Now, in this panel on the right, increasing age at menarche was associated with increased risk of gallbladder cancer, intrahepatic bile duct cancer, and extrahepatic bile duct cancer, but only among Asian women, represented here by the white circles, not among non-Asian women, represented by the black circles. So to summarize this section, there is biologic support for the finding between parity and gallbladder cancer because pregnancy increases the risk of developing gallstones due to high levels of estrogen and progesterone in the second and third trimesters of pregnancy, and that both leads to increased cholesterol saturation of the bile and reduced emptying of the gallbladder, and both of those factors affect your risk of developing gallstones. The association that we saw for intrahepatic bile duct cancer suggests that estrogens might play an important role in the development of cholangiocarcinoma as well. Now, our result was somewhat borderline and needs additional evaluation, but I think this has also been supported by work from Catherine McGlynn and Jessica Petrick, where they found circulating levels of estradiol were associated with increased risk of intrahepatic bile duct cancer. Later age of menarche was associated with an increased risk of gallbladder cancer, intrahepatic bile duct cancer, and extrahepatic bile duct cancer among Asians, but not among non-Asian women. Now, Asian women have a lower BMI than women of other ethnicities, and that could delay puberty onset. And the fact that later of age of menarche was only associated with increased biliary tract cancer risk in Asian women again might suggest that there's potentially a different etiology in Asian compared to non-Asian women. Just to give one example, brown pigment stones are more common in Asian populations than cholesterol stones, and these stones are created due to parasitic infection, for example, through liver flukes. So this difference in etiology also fits in with the descriptive epidemiology, because this sex disparity, that at least 2-to-1 ratio that we see so often in Western countries, tends to be smaller or sometimes may even be reversed in East Asian countries. So certainly it's worth understanding a little bit better these associations and how they might vary in different populations. To give you examples of our future work in BitCap, we want to follow up on the reproductive factors findings, both by using the data that we have in hand to examine exogenous hormone use and biliary tract cancer risk, but also by initiating a new study using biospecimens from cohorts in BitCap so that we can actually measure circulating sex steroid hormones and risk of developing gallbladder cancer and extrahepatic bile duct cancer in postmenopausal women and in men age 50 or greater. Now, another area that we're interested in is pharmaceutical use. For example, aspirin and NSAID use and statins use. And just to give you a little bit of background on why these exposures are of interest, using data from the UK Clinical Practice Research Data Link, Sarah found that it seems like there's actually a beneficial effect on survival among individuals who use aspirin after they're diagnosed with biliary tract cancers. And you can see that here from these survival curves. So here the gray lines indicate individuals who used aspirin after their diagnosis, and the black lines indicate individuals who never used aspirin. And this black vertical line shows you survival at 12 months. And what you can see is that there's about a twofold or sometimes even greater improved survival among aspirin users among non-users. So this is certainly something that's very interesting and something we're working on following up on. Now, incidence is a little more challenging to evaluate because aspirin is available over the counter. So here, when we're looking at survival, we think we might be capturing that exposure a little bit better among cancer patients who are already coming in for many things and may very well have a prescription for aspirin. But among your standard person, you tend to just get aspirin over the counter, and that can be hard to capture in a claims database like CPRD. So one way to address this challenge is by evaluating self-reported data, and that's something that we can do in BitCap. To provide a little more impetus for the statins analysis, Ziwei Liu is a postdoc in our group who previously evaluated statin use in biliary tract cancers in the UK Clinical Practice Research Data Link. And as you can see here, he found that current statin use was associated with a reduced risk of developing biliary tract cancers, as was increasing number of prescriptions and increasing cumulative dose. But we didn't really have the power here to look specifically at each anatomic site in the biliary tract. So this is something where we hope to get more cohorts to provide statins data so that we can really dig into this question and look more carefully at each anatomic site. The findings are important because it suggests that if this association holds up, statins potentially could be used for biliary tract cancer chemoprevention in high-risk populations. For example, patients who have primary sclerosing cholangitis. Now, BitCap has given us an unprecedented opportunity to evaluate classic cancer risk factors and the risk of developing cancer across the biliary tract. But we've also been investigating associations with previously unsuspected risk factors, such as aflatoxin. Aflatoxins are a family of toxins produced by certain fungi that grow on corn, peanuts, other ground nuts, and spices. And we've known for some time that aflatoxin can lead to the development of hepatocellular carcinoma. But there's actually several lines of evidence to support the biologic plausibility of aflatoxin causing gallbladder cancer, including experimental, animal, and occupational data. And in Chile, where the rates of gallbladder cancer are very high, recent evidence has identified aflatoxin contamination in food products, such as red chile peppers, whereas the prevalence of strong cofactors for liver cancer, like hepatitis B virus, are pretty low. And that might help explain why liver cancer rates are low in Chile, but gallbladder cancer rates are high. Now, even with all of this interesting background data, up until recently, nobody had ever looked directly for an association between aflatoxin and gallbladder cancer. So we used data from a pilot study that I conducted in preparation for the Chile Bills cohort, and this included recruiting cases from Santiago, Concepcion, and Temuco with age, sex, and site-matched controls. And my former postdoctoral fellow, Leticio Negeda, initiated this work, working with John Grootman at Johns Hopkins University to test samples from 36 gallbladder cancer cases, 29 gallstone controls, and 47 population-based controls. And here you can see the actual levels of aflatoxin lysine adducts among population-based controls, gallstone controls, and gallbladder cancer cases. And in this figure, each bar represents an individual person, and this red line across the bottom indicates the lower limit of quantification. So everybody who's below this red line had undetectable levels. And what you can see here is that gallbladder cancer cases were both more likely to have aflatoxin detection and more likely to have higher levels of detection. So just to put that in context, the odds ratio for aflatoxin detection among cancer cases compared to population-based controls was 13. So cancer cases were 13 times more likely to have detection, and compared to gallstone controls, that odds ratio was 9. So these are really strong associations, very striking, super impressive, but keep in mind, based on a really small number of people and just the first study to ever really look at this. So we needed a place to replicate these findings. And to do that, I turned to the Shanghai Biliary Tract Cancer Case Control Study, which is a population-based case control study conducted by Ann Singh when she was here at NCI. And she ran this study in the late 1990s to the early 2000s, recruiting over 600 biliary tract cancer cases, most of whom had gallbladder cancer, over 1,000 patients with biliary stones, and about 900 healthy controls. And then again, I worked with John Grootman to test samples from 250 gallbladder cancer cases, most of whom had gallstones, and that's what our primary analysis was based on, and then 250 gallstone controls. We found that 32% of the gallbladder cancer cases had detectable levels compared to only 15% of controls. After adjusting for age and sex, the odds ratio for detection of aflatoxin was 2.7. And then if you limit that analysis to people who actually had detectable levels, the odds ratio for the highest quartile versus the lowest quartile was 7.6. And just to illustrate that, in this figure you can see this dotted line, which represents the gallbladder cancer cases, is shifted up from the solid line, which represents our gallstone controls. We wanted to get a sense of to what extent these gallbladder cancer cases might be due to aflatoxin. So we calculated population attributable fractions. Now, keep in mind this is based on 2 case control studies, and really to calculate population attributable fractions, you need prospective data. So do not take this as truth. This is rough and dirty, just to get just kind of a general sense of perhaps what we might expect. So in Shanghai, the prevalence of exposure in our gallstones controls was 15%, and the crude odds ratio was 2.7, yielding a population attributable fraction of 20%. In Chile, the prevalence of exposure was 24% in gallstone controls, with a crude odds ratio of 5.6, leading to a population attributable fraction of 52%. And I think this is interesting because although this result from Chile is based on small numbers, so you have very wide confidence intervals here, in Chile, nobody knew that aflatoxin exposure was a problem, really, until we did this work. And that means that nobody's been trying to reduce the exposure in this area. That's important because in China, people have really been actively working on reducing aflatoxin exposure for a number of years. So to put that in context, gallbladder and biliary tract cancer mortality in China decreased by 22.5% from 1990 through 2013. And as you can see from this figure, there was this corresponding decrease in aflatoxin levels in this area called Qidong, which is right across the river from Shanghai. As you can see in this area, the median levels of aflatoxin lysine adducts dropped from 19.3 picograms per milligram to being undetectable in 2009 to 2012. And so it's important to keep in mind that in the Shanghai Biliary Tract Cancer case control studies, the samples that we collected were right in the middle of this big drop-off in exposure. So we know that there's some exposure misclassification in this study because the aflatoxin lysine adducts only measure about 3 weeks worth of exposure. So we could very well have some cases who were exposed back here in the early 1980s, and by the time we took their blood in the late 90s and earlier 2000s, they no longer had that exposure, but the exposure that they had back in the 1980s may have led to molecular changes that actually contributed to the development of their cancer. So this is something that's important to keep in mind for the interpretation of this study and also for thinking about what prospective studies we might use to try to better understand these findings. Now, Chile is a little bit of a different situation because, again, here in Chile, we've had no idea that aflatoxin was a problem until recently. So it's possible that there might be a higher proportion of cases that are due to, or at least partially caused by aflatoxin in Chile than in other areas, than in places like Shanghai, China. The next logical step is to evaluate whether there's evidence of temporality, or did the exposure occur prior to people developing gallbladder cancer? And to try to address this question, I turned to the Shanghai cohort study. And this study enrolled over 18,000 men aged 45 to 64 from Shanghai, China, from 1986 to 1989. And just to quickly go back to that figure, that means they were enrolling right in this area. So there might have started to be a little bit of a drop-off in aflatoxin exposure, but still pretty high levels of aflatoxin at the time that these blood samples were collected. Gallbladder cancer cases were identified through the cancer registry and annual in-person interview until 2017. And again, I worked with John Grootman at Johns Hopkins University to measure the serum aflatoxin lysine adduct levels in 84 gallbladder cancer cases and 168 matched controls. In preliminary analyses, the odds ratio for detection was 1.9, so those with detectable aflatoxin levels at baseline were more or less two times more likely to develop gallbladder cancer over the course of the study. But as you can see, this confidence interval is pretty wide, so this estimate is not very precise. And it's also important to keep in mind that the median time to diagnosis was 18 years. That's a long time after baseline, and it's not necessarily clear if associations or if exposures, one second, okay, sneeze is gone, I think, sorry about that. If exposures at baseline might actually impact cancer that develops 18 years later or 30 years later. So we wanted to get a sense of whether the association might vary by time to diagnosis. And we found that restricting to cases who were diagnosed within 15 years of baseline led to odds ratios of just over 3, whereas the odds ratios for cases who were diagnosed 15 or more years after baseline was just over 1.5. We think that given that aflatoxin levels have been dropping in China due to these concerted aflatoxin abatement efforts, the stronger effect for cancers diagnosed within 15 years of blood draw likely reflects the higher exposure in those early years. Now, previous work from China has shown that as aflatoxin exposure dropped, the rate of liver cancer also dropped, and that decline was not due to introduction of the hepatitis B vaccine. And I think that's important because it highlights the potential impact that aflatoxin abatement efforts could have on the public health burden of gallbladder cancer in high-risk areas that have aflatoxin exposure. And in fact, this work has already had an impact. So I presented the results of these case control studies from Chile and from Shanghai to the Chilean Ministry of Health in October of 2016. And following that presentation, the Chilean Ministry of Health then went out and tested a bunch of Merkin spice blends which includes the red chili pepper previously shown to be contaminated with aflatoxin. And after testing for mycotoxins, they actually ended up pulling one brand off of the shelves in January of 2017, which I think really demonstrates the seriousness with which the Chilean government takes these findings and the import of this work. So taken together, we saw strong associations between aflatoxin and gallbladder cancer in case control studies from Chile and China. Preliminary cohort data also supports that association, and I think it suggests that aflatoxin abatement might help reduce the public health burden of gallbladder cancer in high-risk areas. And just to bring this back to the US, as we talked about earlier in this talk, indigenous populations in the US are high-risk populations. They have very highly increased risk of gallbladder cancer, and I wonder if part of that increased risk might be due to aflatoxin. Previous studies have shown that marginalization is associated with higher exposure to aflatoxin, which could be in part due to consumption of self-produced rather than commercially produced foods. Because while commercially produced foods are treated and stored to prevent fungal growth and are also tested to ensure low levels of aflatoxin, rural, marginalized populations are more likely to eat food that they grow themselves rather than commercial products, and therefore may be at increased risk. They might also be exposed to aflatoxin through inhalation of contaminated feed for livestock. High exposure to aflatoxin has been demonstrated in indigenous and low socioeconomic status populations in Mexico and Africa, so it leads me to wonder if that could also be a problem here in the US. One way to gain insight into the potential role of aflatoxin and other factors is by investigating the molecular characteristics of the tumor itself. To give you an example, in a collaboration that we developed over many years with investigators here at NCI, including Ben Zhu, who is a tenure-track investigator in the biostatistics branch of DCEG, and a number of other investigators from the University of Copenhagen in Denmark, we profiled the mutational landscape of gallbladder cancer tumors using whole exome sequencing in 60 tumors from NCI studies, along with 32 publicly available cases that had paired tumor and non-tumor DNA for a total of 92 cases. And then we conducted a validation on a set of 98 additional gallbladder cancer cases with targeted sequencing for those top mutational hits. And in a subset, we conducted RNA sequencing, copy number array, and the epic methylation array, so that we could get a more comprehensive picture of the molecular characteristics of gallbladder cancer. This figure shows you the mutational landscape of gallbladder cancer tumors based on whole exome sequencing. And the main things that I want you to walk away with here are first, that overall the mutation rate was really pretty low, and that TP53 was the most commonly mutated gene. Now, what's interesting is that even though TP53 was the most commonly mutated gene, we didn't see the R249S mutation in TP53 that's typically associated with aflatoxin-related hepatocellular carcinoma. We also looked at the single base substitution mutational signatures, and that's another way to kind of look for evidence of aflatoxin exposure. We found that about 40% of the cases that we evaluated did have this aflatoxin signature, and these mutations tended to be present in the majority of cancer cells, suggesting that aflatoxin had an effect on the early development of the cancer. So one other note about the R249S mutation in TP53 is that some recent work suggests that that mutation might actually be driven by hepatitis B infection. And in this group of cases, there was a pretty low prevalence of hepatitis B infection. So that could be why we're not seeing that specific mutation in TP53, but we are seeing evidence of aflatoxin exposure using this broader mutational signature. Another finding of note was that ApoBec-mediated mutational signatures were more common in tumors with higher mutational burden. And the ApoBec cytosine deaminases protect from viral infections, which makes it kind of interesting. We then explored molecular subtypes identified through clustering tumors by genes whose expression was associated with survival. We had this data from 47 tumors. we identified a 95 gene signature that stratified all of our gallbladder cancer cases into 3 subtypes. Subtype 2, which had good survival, and then subtypes 1 and 3, which had poor survival. Now, these 2 poor survival subtypes were associated with adverse clinical pathologic features, immunosuppressive microenvironments, and T-cell dysfunction, while the good survival subtypes showed the opposite features. And just to give you one example, we used the TIDE immune dysfunction and exclusion, or TIDE algorithm, tumor immune dysfunction and exclusion, TIDE algorithm, to evaluate the predicted immune cell functionality. And one of the things that we found was that the potential for T-cell exclusion was higher among the 2 poor survival subtypes compared to the good survival subtypes, suggesting that the T cells are getting into the tumor more readily in this good survival subtype group. We then evaluated the digital H&E images and found that subtype 2 had greater immune infiltration compared to subtype 3. So it was good to see that verification of what we had seen based on the RNA sequencing data. Taken together, these findings suggest that gallbladder cancer survival may be dictated more by microenvironment than by something specific in the tumor itself. While these findings are suggestive, again, this is just one study and it's based on a pretty small number of cases. So one place that we hope to confirm and expand on these findings is in the Chile Biliary Longitudinal study. Chile Bills is a cohort of women with gallstones that is going to allow us to address this key question of what are the factors associated with the development of gallbladder cancer among individuals who already have gallstones. Chile is a great place to address this question because it has a very high prevalence of gallstones and among the highest rates of gallbladder cancer in the world. These rates are particularly high in southern central Chile, where there's a high predominance of individuals with Mapuche Amerindian ancestry. There are also a number of important opportunities to evaluate exposures of interest. For example, genetic admixture, Salmonella enterica ceravariant typhi, heavy metals, and aflatoxin exposure. And another important point is that there's really an urgent need to find ways to identify high-risk people so that we can prioritize them for cholecystectomy. That's important both because survival for this cancer is very poor, but if you can catch it early, survival increases from less than 20% to up to 90% or even more. And in Chile, there are so many people who need cholecystectomies, there aren't enough surgeons to do the surgeries. So the government prioritizes people according to age, but in something of a surprising way. They actually prioritize younger people, age 35 to 49, with the thought that these people don't have cancer yet, so we'll take their gallbladder out now and they'll never get it. But of course, that means that the older individuals, people age 50 and above, they have to wait. Sometimes they have to wait for years. And these are the people who are already at increased risk of having or developing gallbladder cancer. So this is something that we hope we can address in Chile Bills to find more biologically based ways to help with this stratification of the cholecystectomy waiting list. But that said, the primary aim of Chile Bills is to investigate the epidemiologic and molecular predictors of high-grade dysplasia and cancer in the gallbladder. And some of the things that we're especially interested in include inflammation markers, ultrasound characteristics, and obesity, diabetes, and metabolic syndrome. Another major aim is to characterize the natural history of disease. In other words, what factors affect the transition from chronic cholecystitis to intestinal metaplasia, from intestinal metaplasia to low-grade dysplasia, and then finally from low-grade dysplasia to high-grade dysplasia and cancer. And we have extensive biologic specimen collection in Chile Bills, which I think will really facilitate these efforts, enabling the in-depth evaluation of the molecular epidemiology of the natural history of disease. Creating this cohort involved first identifying 29,000 women aged 50 to 74, and then screening over 19,000 women with ultrasound so that we could identify 4,726 with gallstones. We're following these women for 6 years with study visits every other year, and we expect that by the end of the study, we'll probably identify about 180 to 200 gallbladder dysplasia and cancer cases. Right now, we are finishing up the year 2 visit with 84% compliance, and we are also conducting the year 4 visit with 86% compliance. So far, more than 1,600 women have gone to surgery to date. To give you a sense of the baseline characteristics of this cohort, as you can see, 55% were age 50 to 59, 53% had 8 years or less of education, about a quarter are of Mapuche Amerindian ancestry, about 60% are obese, 25% have diabetes, 6% have cardiovascular disease. Now this last point is important. So about a third of the cohort had ever experienced having biliary colic at baseline, which means that most of the women in this cohort actually were asymptomatic and that they had never previously had biliary colic. That's important because the vast majority of people with gallstones are asymptomatic. They don't know that they have gallstones. And that's something that we really wanted to achieve in Chile Bills, to be able to get a cross-section of individuals with gallstones, not only those who are asymptomatic. So this is really encouraging because it shows us that we've accomplished our aim in that sense. 100% of the cohort had hepatobiliary ultrasound at baseline, and of course that's by default because we had to do ultrasound to see if they had gallstones in the first place. But 99% also had a physical exam, 96% gave a baseline blood sample, and 99% gave a baseline questionnaire data. It took us a little while to get up and running with fresh frozen tissue collection, but now we're capturing the majority of participants who go to surgery so that we can get those fresh frozen tissues. And here you can see the surgical samples collected through January of 2020. In addition to these, we're getting FFPE tissue on 95% of participants who go to surgery, and we have completely sampled gallbladders for about 75%. So we're really making some progress with these sample collections. Chile Bills is particularly valuable because this is the first well-characterized gallstone cohort in a region with high risk of gallbladder cancer. And there's a high prevalence of understudied exposure. We're conducting extensive biospecimen collection so that we can both really investigate the natural history and biologic plausibility of exposures and conduct extensive molecular efforts. We are also collecting imaging data over time. So we're getting ultrasound images, we have it from baseline, but also year 2, year 4, and we will have it from year 6. And we have digital H&E images for those individuals who go to surgery. As you can see, Chile Bills is a truly unique study that will allow us to gain new insights into the etiology of gallbladder cancer, as well as generating risk estimates for the transition from gallstones to gallbladder cancer in a high-risk population, and potentially to enable translation of some of these findings into a positive impact on public health. To wrap everything up, what are some of the lessons that we've learned from studying these rare cancers? Well, first, pooling projects like BitCap can enable prospective evaluation of rare cancers in a way that's just not possible for any single cohort. Second, studies in high-risk areas might help us identify novel associations like aflatoxin, which we first identified in Chile. Third, intense integrative molecular analysis of a relatively small number of tumors can really help identify important leads. And finally, geographic hotspots facilitate etiologic studies and identification of targets for cancer prevention and early detection. Of course, none of this work was done in isolation. There are a number of people involved, and I'm sure they're not even all listed here, but I would like to acknowledge the many people, expert colleagues and collaborators from all over the world who have contributed to these efforts. And I'd particularly like to highlight my fellows and my former fellows who were really vital to the work that I presented today. I'd like to thank them and also to thank you for your time and attention. And with that, I will stop and take questions.
So that was an amazing talk. They don't let me get on the video, sorry. But, let me tell you, before we go into the specifics, because, we all doing these virtual talks and, you always miss audience. So, you know, I want to tell everybody what I noticed and wrote down during this talk. So there are 80 people online, and I'm not going to give you names, but the people that listen here come from Georgetown, the Global Liver Institute, from UCSF, from Mayo, from Harvard, and from the Cholangio Foundation. You know, and these are just a few that I where I noticed the names and could recognize them. So, you know, I think it speaks for itself that, you know, people are interested in this type of meetings. And congrats, that was an amazing talk. I mean, you know, who else can say that their research has so much impact that you actually take the spice out of a supermarket in the entire country? You know, I mean, you know, that speaks for itself. So, you know, if anybody has a question, if you just put a message in the chat, you know, we can ask you to say something, and you can start your microphone. And before we and people do this, you know, there's actually Chris Lofredo from Georgetown already has a question.
Hi, Jill. Thank you so much. It's great to see you, and congratulations on this wonderful epidemiological work. To see somebody overcoming these hurdles to put together consortia and to start that very impressive study design done in Chile, just congratulations. It's really very, very great. I do have a question about that Chile design. I think it gives you the opportunity to assess some of the 4Fs you talked about, and their role both in etiology and in progression of disease. So you posed this model where we wonder if the same factors that lead to the gallstone then influence the further progression on towards cancer or dysplasia. And I think now with the study design, if you have the data, you certainly have the biomarkers maybe that you could look at what changed and what was continuous. But if you have the data, you could evaluate things like changes in body composition between those time periods. You could look at whether people lost weight, for example. You could look at whether new exposures came on, whether somebody quit smoking. That, of course, is going to require a somewhat more sophisticated data analytical plan, something like sensitivity analysis or mediation analysis is probably more what I mean, or even path analysis, something like that, to account for that sort of complexity. But have you thought about the opportunity that you would have to sort out some of those intriguing questions that you posed early on?
Yeah, Chris, that's a great question. Thank you so much for bringing it up and pointing it out. I think you're exactly right. That is exactly the kind of thing that Chile Bills allows us to do that really no other study has been able to do. I think parity, for example, is a great one. You know, we know parity is associated with gallstones, but is it only the gallstones, or does it do something additionally? So that's something that we'll be able to evaluate in Chile Bills. Also looking at body composition, we are getting height and weight, and we have waist to hip measures, and we have that physical examination. I'm not sure we have it this year because of COVID, but at least for year one and for most of year two, we have that. To the extent that we can get it safely, we will continue to get it. So that is part of the hope, that we can kind of look at those changes over time. And certainly for any biomarkers, so we can look more directly at hormones, for example, just actually measure the hormones at baseline, at year 2, at year 4, at year 6, and see how those things change. One thing that I'm really interested in, it will take some careful work and planning with our statisticians because the numbers get small. But one thing I'm very interested in is, to what extent might the association with obesity be through inflammatory pathways? And can we really kind of dig into what that mechanism is? So looking at that kind of mediation, I think, is really important and valuable, and something that we certainly want to do.
Great, thank you very much.
There are a number of questions, so we have 6 minutes left, so you can give 2 minutes per question. So why don't we start with Katie Kelly from UCSF, actually.
Oh, hi. That was a wonderful talk, Jill. I had a quick question, which I wrote in the chat as well, about the pigment stone observation and whether you've looked at hemoglobinopathy, obviously more prevalent in Asian populations, and whether hemoglobinopathy is associated with cholangiocarcinoma. I saw a couple small studies from 30 years ago, but not much in a PubMed search that I looked just now.
Yeah, we haven't actually looked at that. That's a good idea. It's something we could follow up on, but it's not something that we've looked at yet. Thank you.
Shin?
Oh yeah, since I don't have much time, I'm just going to ask one question very shortly. Is the size of the gallstone matter in terms of producing different kind of information that more driving, the aggressiveness of the tumor?
Right. So it's not entirely clear. There have been studies that have looked at the size of the gallstone. I think volume of the gallstone has been more consistently associated with increased risk. But I don't know that anybody has actually looked at how the size or the volume of the gallstone is related to inflammatory factors. And that is something that we can do in Chile Bills. Size is challenging because the stones are often crushed through laparoscopy. It's late, guys. Laparoscopy to get the gallbladder out. But we are getting volume and we're measuring the largest stone where possible. So it's something that we can follow up on.
Jake DeYoung from NIDDK.
Yeah, hi, Jill. I agree with Tim. It's a great talk. I wonder whether you thought about the sort of the role of infection in cholangio, in gallbladder cancer, especially we know, for instance, colonucleus is a well-known cause of gallbladder cancer, but there may be other unknown infections that we really don't know about that may be associated with gallbladder cancer. Can you comment on that?
Yeah, I think you're right. There's been a lot of interest in Helicobacter species, for example, and how they may be related. I think the problem there has been measurement. Especially for the species rather than pylori, it's just hard to measure them well, which has for me been kind of the hang up in trying to address that question better. We did try to do some 16S sequencing to get at microbiome using bile samples that we had from the Shanghai study. But we ran into trouble with inhibitors. We weren't able to really amplify the bacterial DNA, and it wasn't clear if it's because it just wasn't there, or if there were problems with the specimen type that we were working on. I think it's an important question and something that I would love to work with somebody on more carefully to really be able to develop the methods to test bile and understand what is there, what does the microbiome look like within the gallbladder.
Great. Well, we can talk more later. I know we're supposed to meet after your talk. So.
Perfect.
I guess if you have still time, Tim, I have a quick comment. And,
all right.
I think this is about the aflatoxin comments for Qidong study. I think that we see a reduction of the aflatoxin, at the same time reduction of HCC incidence in that area. But this study actually confounded largely is due to a changing diet, because from early stage of the different diet now to a rice diet. As a result, and actually the nutrition effect actually changing the incidence. That has been pretty largely accepted instead of aflatoxin. So.
Got it. Yeah, I know that there was a change in that they allowed rice into Qidong, and that made a major difference.
Plus, not only the rice, but also increasing the nutritional factors. For example, people become more wealthy and they have a better diet instead of early those kind of corn-based diet.
All right, so again, thank you everybody, and let me allow to announce our next speaker on April 22nd is going to be Bernie Redd, our radiologist here from NIH. And for those of you who don't get enough, there is a number of meetings coming up. The Cholangio Foundation has their meeting March 31st. There's a FASEP meeting, and I think HCC tags. So lots of opportunities to learn about hepatobiliary cancers. Bye.